Great. Well, so thank you all again for coming to my my second lecture here in this set of talks on spatial population genetics. I know that you've had many talks already today and you're probably pretty tired. So I appreciate you coming. And as I said before, welcome questions both from experts like Kavita, but especially from the students and the postdocs or whatever in the audience. So in this second talk, I'd like to explain this phase diagram over here. It's a kind of phase diagram which depends on parameters, alpha and beta, parameters that tell us something about how two different genotypes, like the green and the red genotype in the top picture, how they interact with each other. And as I mentioned briefly last time, there are lots of ways that they could have some kind of interactions. One is simply that one species outcompetes the other and reproduces more rapidly. That's survival of the fittest in a strict Darwinian sense. This picture here of a bacteria called Pseudomonas aeruginosa with two different genotypes. In fact, the two strains are neutral and they don't have any fitness advantage, but nevertheless, they segregate at the frontier, which is an interesting phenomena that we'll talk a little bit more about today. But if I make things more sophisticated, move away from this Darwinian paradigm, there are parameters alpha and beta, which I will tell you about, that control more sophisticated kinds of interactions. And briefly, although this is the main subject of the talk, when alpha and beta are both positive, the two bacterial or yeast species cooperate and provide shared public goods, sort of like the idealized model of free trade between two different countries that exchange things that the others don't have to mutual benefit. On the other hand, down here for negative alpha and beta, as we'll see in detail, there's antagonism. That's what these skull and crossbones are intended to indicate. They're fighting each other. If one is antagonistic and the other is happy with this little heart here, here's the opposite situation where one is very generous and the other is very nasty. That's this Darwinian paradigm. So in the second quadrant and the fourth quadrant, it turns out, as a function of these parameters, which I will explain to you, we have a Darwinian situation where one, for whatever reason, one species simply outcompetes the other. But especially interesting things happen, as I'll mention at the end of the talk, when they're both antagonistic, because in that case, in space, we have a genetic analog of a line tension that tends to want to minimize boundaries, just like line tension plays an important role in physical chemistry, for example. So that's where we're going today. And just to remind you where we were last time, please recall this experiment where I spotted down 5% of a red genetic bacterial species, Escherichia coli, and 2%, and looked at what happens. So this homeland that is the starting condition. Here's another homeland, and then there's this outward migration. And this was to illustrate this phenomena of gene surfing. And here's an example where, as you may recall, there was somewhere down around here, we can locate in space and time a kind of bacterial Eve whose descendants all have this red color, this red fluorescent protein that they express. Otherwise, the red is neutral. with respect to the green. These were grown in a dark refrigerator or a dark incubator, usually it was an incubator. And so there was no light most of the time they're growing. We would pull them out and just take a picture of them. So they're very, very neutral. But here's a case where this red-green boundary random walked a little bit by accident to the right. This other one random walked a little bit by accident to the left, producing this, this sector, which once it gets far enough apart, is going to persist for a very, very long time. And that's an example of successful gene surfing. Here's an example of unsuccessful gene surfing, where the red strain was squeezed out eventually. And I see a question from Sayantan. Please ask.
Yes, in the past, where the red is the surfing, we can see there is a light red patch I can see on the top part, above that,
like here?
Above that, on the outer ring.
Okay, okay, yes.
On the
okay.
Outside the green border, green circle, I can see a light red patch. So is that because of the bacteria? Or
This light red band.
Yes. That's what.
Yeah, I think that's an artifact of. That's a great question. I think that's an artifact of the optics, and it's something to do with the camera. As far as we know, that's not due to bacteria. But excellent question and a good observation.
Yes,
Any questions right now? Okay, thank you. I will have to think more about that, but I don't think that's bacteria. I think that's an artifact of the optics in this case. Okay, so I mentioned this very briefly in the question period. You know, the nice thing about these experiments is that, unlike the human range expansion out of Africa, we can repeat the experiment 20 times and get statistics and check our theories and so forth. And indeed, we can imagine this as, in the words of my colleague Stephen Jay Gould, replaying the tape of life. And so here are, as I showed at the very end yesterday, 3 range expansions. And R0 is the initial radius. And if I look at this one here, where I have, you know, perhaps 8,000 different seed E. coli, each having a heritable red or green color. Here, maybe I had 4,000, here I have 2,000. So the homeland is coarser, but there's a kind of universality because they always sector at long times. And one question we could ask is a kind of inference question. Given information on the boundaries of these sectors, can I infer the size of the homeland? That's a kind of, you know, simplified version of the questions we'd like to ask about human migrations. From present day data, can we infer what happened in the past? And for this problem, it turns out you can do it. And what you have to do is count the number of sectors. These sectors are random, bounded by random walking boundaries. And those boundaries are inflating away from each other because the rim of this bacterial colony is growing linearly with time. And the boundary between the genetic boundary between the sectors is undergoing a kind of square root of time random walk, at least approximately. And so once the colony gets established, the inflation at the frontier outruns the random walk annihilations. Like here, there's annihilation, which kills off this colony here, or this red sector. And by counting sectors at long times and using other data at the frontier, you can infer the size of the homeland. In fact, here's the formula. It's a pretty simple formula to derive. The hard part was getting the square root of pi over 2. But what this says is that the number of sectors that survive, which we can count, doing 20 runs or something like that, is related to the initial radius of the homeland by multiplying by the velocity of the range expansion. It settles down to one of these Fisher wave velocities, which we can measure, and then also related to a diffusion constant that controls the wiggling, describes the amount of wiggling of these sectors. So we can measure that kind of root mean square wiggling of this walk in the angular direction as it goes out radially. So we measure dW, we measure this velocity, we measure the number of sectors at the frontier, and we can invert this equation and get the radius of the homeland. So there's a theory that does that. I'm not going to go into it in detail, but it actually seems to work. This is because these are neutral strains. This is survival of the luckiest, and to ensure that there isn't some accidental mutation. They occasionally happen, but they're rare in these E. coli. What you can do is go and scoop out a little bit of the red that you might think is the result of an actual mutation, as opposed to genetic drift, giving red a selective advantage, and then recompete it with the green. And it doesn't have any signs of actually being a favorable mutation. This is just, as I said, survival of the luckiest. One of the nice things about doing experiments and comparing them with theory is that there are sometimes surprises. And so here is an example of a surprise. This is a somewhat different strain of bacteria. And you'll notice that we get the sectoring. This is the homeland, but it's chiral. It has a kind of a clockwise rotation, on average clockwise rotation to the sector boundaries. We think that might be related to the chirality of the peptidoglycan cell wall of the E. coli. That's a chirality which occurs on the length scale of a micron. Here we're seeing it on the scale of several centimeters. So it is like 10,000 times bigger scale, but we see it. My original thought was maybe it was because we did the experiment in the Northern Hemisphere, but probably not. Probably not affected by Coriolis forces. Maybe if we did it in Sri Lanka, we'd be very close to the equator, and then we get equal numbers of left and right-handed. But I don't think so. I think it's a feature somewhat like a cholesteric liquid crystal of the chiral rigid cell walls of the bacteria. But anyway, that remains to be seen. But it's nice to have surprises in experiment because it makes you think. So here's a summary of what's going on, just from the perspective of one of these razor blade inoculations. So the razor blade was touched on this vertical path in this particular experiment, having dipped first the razor blade into a 50-50 mixture of red and green nutrient broth. And then that wet razor blade edge was touched down here to launch a range expansion to the left and a range expansion to the right. Somewhat easier to think about theoretically because there's no radial inflation to worry about. And under these circumstances, if these strains are neutral, and we think that they are, randomly green or red will take over all along this razor blade, and eventually the left will all become completely red or all become completely green. So without that inflationary modification, we have a definite outcome. And as I mentioned, what we think is going on at the frontier is we start with a mixture of these organisms. There aren't so many of them at the foot of the organism. And so as it moves forward in time, it's almost like a serial dilution experiment where you would take this test tube full of green and red organisms and then pipette over to fresh media, pipette again, pipette again. And so each generation time is almost like a serial diluting pipette experiment. And now, in this context, I want to do the simplest thing to make the genetics more richer, and ask, how do we put in a Darwinian selective advantage that would act on top of this genetic drift? Darwin, I don't think, knew about genetic drift and survival of the luckiest. Of course, his brilliant observation about survival of the fittest is what we're now going to address. So I'm going to do some more equations. Pretend, if you will, that I'm at a blackboard, and I'd like to talk about how we incorporate selective advantage into these kinds of theories. And I'm going to leave out right now stochasticity, and the discreteness of the cells, and just imagine a world where I have, let's say, green offspring, and they, at each time step, they're capable of reproducing a total of WG new green organisms. And similarly for the red, at each time step, they will produce W sub R organisms. And then we can talk about red and green fractions. And I'll assume here that the green fraction is larger, the number of potential offspring from which the next generation will be selected. And I'll assume that there's a fixed carrying capacity, so the number will be fixed. But these are potential offspring. But the potential green offspring will outnumber the potential red. So there is a Darwinian selective advantage, and this green is preferred. So how does that come in to the mathematics? And so if F is this green fraction at a certain time, then 1 minus F sub G will be the red fraction. And what I want to calculate now is the change in the green fraction after one generation, after allowing all the bacteria to reproduce. This is a simplification, assuming that they all reproduce in tandem. In reality, the cell division events for bacteria are staggered in time. But if we coarse grain a bit and test against simulations and do more fancy mathematics, that doesn't seem to matter. So let's imagine we have discrete time steps, discrete generation times. As Kavita said, that's about 20 minutes for E. coli, or 40 minutes, depending on the strain. And I'll look at the change in this fraction going forward one generation time. So if I start with a green fraction, each one of which produces WG offspring, then that gives me this numerator here. In the denominator is the total number of organisms. And so that's WG times FG plus WR times 1 minus FG. That's the number of red. And then I subtract that from the FG I had before. So this is the fraction in the new generation, F sub G of T plus tau G. And this is the fraction I started with. And then if you just simplify things a little bit, what you'll find is up in the numerator, WG minus WR times F times 1 minus F. That's probably the most important part of this equation. Down here in the denominator, I have something a little more complicated, but we're going to be assuming that WG minus WR is small. It's positive, let's say. So there's a selective advantage for the green. And so if this is small, 2FG minus 1 is bounded. FG can't escape the bounds 0 and 1. So this thing goes between 1 and minus 1, let's say. And so this is small. So if I neglect that, and then just take the differential limit, or small time steps. So we assume, as I said, that the difference between these WGs is smaller than their average. The result is this sort of classic equation of Darwinian population genetics, which says that dfg dt, as indicated by this numerator, is a number, a positive number, which we call the selective advantage. It's 1 over the generation time, but then importantly, it has WG minus WR in the numerator and then the average in the denominator. So there's a selective advantage. And the solution of this simple differential equation is just what Darwin would like. It says that the green fraction, as a function of time, grows exponentially at short times, but then at long times it saturates. And here it's saturating at 1, meaning that the green fraction completely takes over and squeezes out the red fraction at long times if there is a selective advantage. So that's this classic picture. And let me throw caution to the winds and attempt to draw a graph here. So this is, time is this way. This is f itself, and it sort of looks like this. It's the sigmoidal curve. So that's a classic result in a population genetics context. Just like the populations go up to their carrying capacity, this fraction of green goes up to its maximal value if it has a selective advantage. In that case, the maximal value is 1. And so we can now ask, okay, let's assume this is happening in some spatial context, and we can add diffusion to the fraction. So the fraction is diffusing around in one-dimensional space. And I hope that if you were at the lecture yesterday, you recognize this as a Fisher equation. And it's a Fisher genetic equation, and it describes how, in fact, one organism can take over from another in space. And I see that Sayantan has raised a hand, so please ask your question.
Yes, sir. I probably missed in the expression of S, the tau g in the denominator. It came from the df tau t plus fg t plus tau g. Is it?
Yeah, thank you. So thank you for that question. So yes, where it comes from is doing a Taylor series expansion of f sub g of t plus tau g. And so I'll get tau g times the derivative, and I divide through by tau g, and that's what gives this tau g in the denominator here. It's important because it makes it dimensionally correct. This f is a fraction, so it's dimensionless. 1 over t has dimensions of 1 over time, and so this s has to have dimensions of 1 over time, and that's where it comes from. So thank you. Are there any other questions?
Thank you.
Yes.
Yes.
Yes. Please.
Yep. So this equation is indeed familiar, but I was not aware of this proof, and it's indeed more elegant. But then the question is, like, follow up to the previous one, is why it stop at first order expansion? Like, if we go further, like, what would be the reason to stop at?
Okay. So we could go to second order in time here, and I haven't looked at this problem recently. But I think you can show that if the fractional population change in a single generation is small, if it only changes by 1%, that or less, or some small number like that, then this expansion is good to stop at first order. Because if it's a small change, then basically tau g is like a small parameter, it turns out. And so a term which is tau g squared, which is I think what you're asking about, or tau g cubed, will not matter. So it depends on the problem, but if the change is small, and it will be small if the selective advantage is small, then I think we're good. You can certainly put in corrections, but I don't think they'll change the long time asymptotes.
Thank you.
Yeah. Any other questions?
Yeah, I have a question.
Please,
So,
Kavita.
yeah, so for this genetic wave equation, basically what you did yesterday also,
Yes.
but now we have selection. I was just wondering what if S is a time-dependent object? That means, like, for example, I'm thinking of fluctuating selection. So I can't even start with that sort of traveling wave ansatz, right? So would you know, like, is there even a traveling wave, or if it is, does the speed change?
Yeah, that's a fascinating question, and it is very much related to trying to generalize these models to allow for things like climate change. That is, the environment could be changing with time, and you could even have a change in sign if this S is time dependent. And I think very little is known about that. If S, my guess is that if S is always small and positive, that it's not going to change much. But I would also emphasize, as I'll show on the next slide, maybe I'll do that now, that in fact, a more correct equation, even without a fluctuating S that you ask about, Kavita, will have noise. It'll have a stochastic noise associated with it. And so my guess is if this noise is stronger in its fluctuations than possible fluctuations in the environment, it's okay to take S to be approximately constant. But you're right, in general it won't be. And I think that's a fertile area of inquiry, but I don't know.
So I was actually thinking of on an average neutral, so that it fluctuates about 0 basically.
Okay.
I would think that I'll have to add one more term in the Langevin equation for the, suppose S is chosen from Gaussian distribution, right? So I'll have to add one more fluctuation, and then there'll be a competition between 1/n versus
exactly.
With the fluctuations, or even there could be correlations. So okay, I don't know.
And that's a great suggestion and a nice simplified problem. And I don't know the answer. When you figure it out, let me know. But here we are assuming that the environment is held constant. That's certainly true on these Petri dishes, but in the real world, there are seasonal fluctuations in the amount of sunlight and the temperature, and then there can be long-term fluctuations as well. So we're assuming that the environment is held constant, but that need not be the case. So that, thank you.
Thank you.
Other questions? So anyway, here's this picture, and now I've modified it for genetic drift and selection. So selection is this first term, as we just saw here. And there's a selective advantage, maybe small number like 0.05 or so forth coming in here. That will produce in F of t this sigmoidal curve that I talked about before, that is this particular solution of this nonlinear differential equation. But if you do a simulation using Fisher-Wright modeling or Moran modeling, this sigmoidal curve will have superimposed on it these wiggles. And that can be modeled either in a kind of Fokker-Planck-like way, but also just in terms of an unusual Langevin equation, where I add this noise. And I'm not going to prove this, but this is something that the great Japanese population geneticist Mutu Kumura introduced into this field. And what's fascinating about it is unlike Langevin equations for pollen grains in Brownian motion, the noise itself might be delta correlated, but it gets multiplied by a nonlinear function of f. So f appears not only here, or f times 1 minus f, it also appears here, and then 1 by n, 1 over n, is the size of the population in this binomial sampling process. So that's like 1 over n, as I said, is like the temperature in a simple random walk model. But even then, the analogy is not perfect because there's this fascinating f times 1 minus f. What does that mean biologically or physically? It means that if the population is fixed at 1, then, and there are no mutations, then 1 minus f is 0, and there's no noise anymore. And if I start off with no green strains at all, f is 0, then there's no noise. So the noise, the strength of the noise is f dependent in this parabolic or quadratic fashion. And there's a fascinating world that at least most physicists in my acquaintance don't know about, but it produces this noise, and that can have really beautiful consequences. For example, you could have a selectively favored strain. This is all imagining in a well-mixed test tube, just to keep it simple. And the noise, even though it would like to take off and take over the system in a Darwinian sense, the luckiest, survival of the luckiest comes in because it could fluctuate down and hit this absorbing wall at the bottom. And then it would be all over, even if you had a selective advantage. So, you know, if you've ever watched these crazy Hollywood movies about the X-Men, these mutants with amazing powers like flying or ESP or telekinesis, they might be born, but if a rock falls on them or there's some bad luck, they would just die out, and that favorable mutation would never be expressed in the larger population. So this noise involves escaping from this absorbing boundary condition down here, and of course there's an absorbing boundary condition at the top. So it's a fascinating variant, at least for some of us physicists, on conventional Brownian motion because of this nonlinear term here. And that will come into play later on. So any other questions about these simple range expansions on Petri dishes before I proceed?
Yeah. In the previous slide, it's the follow-up question. Just the
okay, sure.
one. So again, for the case of the fluctuating selection, so how should I go from the Langevin to focal plane if I try to make some progress there? So
okay.
I understand there will be some more noise term which will get added up, gamma prime.
Yeah.
That will be, I mean, how that will be related to the gamma set. Is that will
okay,
be also
so if we take Kavita's suggested problem, her nice suggested problem, there would be an independent, I would replace this S of t by, I would replace S by some stochastic function, if I'm understanding her correctly, S of t, and there would be a gamma of t, and they would be completely independent. So gamma of t is an independent process that's related to the fluctuations and the discreteness of the population. And then S of t here would just be related to the environmental noise. Now, the reason why people, I think, neglect it most of the time is that the time scale for the environmental noise in S of t is much longer than the generation time in most cases. And so through excellent approximation, S of t would be a constant, maybe slightly negative, maybe slightly positive, but there might be situations where it is important. And I'd like to know how to do it. There are, if you look in, there's a review of modern physics article by Korolev et al. That I think I may have sent to the organizers, but you can certainly find it. It's from about 8 years ago, which tells you how to go from these stochastic differential equations to a Fokker-Planck equation, where you would then follow a probability distribution in F. And that's how I would try to proceed. It's not obvious to me that that Fokker-Planck equation would be soluble. And I should say for the experts in the audience, and I think Kavita probably knows this, if you have a multiplicative noise here, multiplying gamma of t, which is what happens, it's kind of, it's noise in the sense that f of t is fluctuating. So there's a multiplicative noise here. You have to be careful in constructing the Fokker-Planck equation, and there's something called the Ito calculus, as opposed to a Hubbard-Stratanovich interpretation of the stochastic differential equations. But that's a detail that you can read about. Other questions? Okay, then I now want to spend the rest of this lecture telling you a little bit about what happens when populations interact in more complicated ways than just a simple selective advantage. Of course, we've already talked about neutral mutations. That's, you know, pretty common. Many mutations don't change, for example, a protein in an essential way. If they just change a codon that still encodes for the same amino acid, there's a degeneracy in the genetic code, then to first approximation, that's a neutral mutation. So those are definitely worth looking at. Selective advantage, that's survival of the fittest with a little bit of noise, superimposed, survival of the luckiest, etc. But I want to talk about more complicated interactions. So here's an illustration of what might be going on in these interactions. This is an experiment I did with Hernan Garcia and Rob Phillips. And what was done was to start off a little red colony and a little green colony and allow them to collide. So here they are dividing. You can see them dividing. This is high resolution, so you can see the red and green colors. And then, boom, the colonies collide. Now you see this wall between green and red. Now we chose these strains to be neutral. And so maybe, you know, if it were like an ordinary chemical physics or something, you'd expect them to mix, but they don't. And they don't because if I think of the red and green bacteria as being like chemical species, they are reproducing. That doesn't usually happen when you mix oil and water. The oil molecules don't begat new oil, and the water doesn't begat new water molecules. And so it turns out that the reproduction is so fast at this interface that it stays demixed. And here's an attempt of red to penetrate the green, but it eventually fails. And so we have this red-green interface. You can see, I hope you can see that there are different brightnesses of the color. This red is brighter than the red out here. And that's because it's terraced, that there's probably a second layer. When these collide, there are excluded volume interactions that squeeze up the red to the second layer or squeeze up the green. What I want to address now, though, is what happens at the border as you're growing out. And as I said at the beginning of this talk, and also yesterday, the red and the green could be doing more sophisticated things to each other than simply trying to out-reproduce. A Darwinian selective advantage would bend this interface to the right or the left, but they could be doing more sophisticated things, and that's what I want to talk about now. So here's an example. These are two strains of budding yeast. So they're a eukaryotic organism where a bud comes out from each mother in their asexual form. They have sexual forms as well, but we'll focus on the asexual form. A bud comes out from this yeast, and that would produce a new daughter cell. And so you'll often see budding yeast depicted in this way. Here's another genetic strain. And just like those bacteria that I showed you in the original range expansions, they've been genetically engineered by Melanie Mueller and Andrew Murray, who's a longtime collaborator in our biology department, to have slight differences. Differences are the following. This blue strain overproduces a crucial amino acid, tryptophan, which it exports and shares with its environment. So it's an altruistic organism in that sense. Leucine, it's called leucine minus tryptophan feedback resistant. So what it means is that it just overproduces and exports the excess. But leucine minus means it can't make leucine. So it has more tryptophan than it needs, but no leucine. So it can't live very easily by itself. And the yellow strain, and also it has a heritable blue marker on it. So it can, you can tell who begat whom in the experiments I'm going to show you. And then this yellow strain has a heritable yellow marker on it, but it's the inverse of this thing here. It overproduces the missing amino acid over here. It overproduces leucine in a feedback resistant way, but it sadly can't make tryptophan on its own. So this shares leucine with the environment, this shares tryptophan in the environment, and the interaction between the blue and the yellow strains is a marriage made in heaven because they need each other. And this is a common phenomena in biology called mutualism, where you know, there are, I think, bacteria in the roots of plants that help the plants take up nitrogen. And so the plants are helped, the bacteria have a place to live, and so there's some exchange in a positive way. So we can do that in the lab and look at range expansions in the presence of this mutualism, where the survival requires exchanging amino acids if leucine and tryptophan are not already present in the natural environment. So how do we describe such a thing mathematically? I'll show you the experiment in a minute, but I won't go through all the details. Maybe I'll use my highlighter here. But the idea is that if we have, again, these reproduction rates of yellow and blue offspring, let's say, during one generation, here's our formula for the selective advantage that we already have discussed, this Darwinian selective advantage. If yellow outproduces blue or whatever. But now I'm going to let these W's be a little more interesting. So as before, we'll let F of X and T now be, say, a yellow fraction. So 1 minus F in a constant carrying capacity environment would be the blue fraction. Then what we'll do is we'll let the yellow reproduce at a background rate. That's what it does in the absence of blue. Maybe it has some very weak ability to reproduce, but because it gets tryptophan from the blue, I multiply the blue concentration by a boost factor in the growth rate, beta. And so if beta is positive, things will grow better for yellow in the presence of blue. And similarly, the blue may have some background growth rate G, which we'll take to be the same just for simplicity, but it gets a boost factor in the presence of yellow, which occurs with fraction F. And so now what I can do is plug into this formula where I have this classic case of frequency dependent selection. So the selection coefficient isn't also actually a function of time here too, because it depends on the fraction, but not environmental fluctuations, just fluctuations in the concentration of the organism. So in a sense, this is a little bit like Kavita's problem. But anyway, if you take these formulas for the fraction of Wy and W sub B, put them in here, and assume alpha and beta are small. So we'll assume alpha and beta are small here. Then you'll find this frequency dependent selection coefficient. It's not just a constant, as Darwin would have assumed in his simplest model. It has a prefactor, which is this alpha plus beta over G. So that's like the S0 that we had in the simple model of selection. But now there's an extra structure which to a reasonable approximation, if alpha and beta is small, there's this extra F dependence. It's linear, but when I put it together with F times 1 minus F, in constructing a kind of genetic Fisher equation, instead of a quadratic polynomial over here, I have a cubic polynomial. And as I say, alpha and beta would be positive in the case of mutualism. And notice that there's a new fixed point on the right-hand side, other than F equals 1 and F equals 0. And that new fixed point is beta over alpha plus beta, which is some number between 0 and 1, and is clearly going to play a role in the dynamics. So it's a new kind of Fisher equation, but with a cubic polynomial here in the reaction term. And you'll see that that leads to some very rich behavior. If I just look at what this, suppose I neglect space now, and just look at a well-mixed environment and ask what's going to happen in a well-mixed test tube, what we would predict to happen from this polynomial by just plotting S0 times F star minus F times 1 minus F, has zeros here and here, and there's a stable fixed point in the center. And so the prediction is, regardless of where I start, depending on alpha and beta, if they're both positive, the system of mutualists will go to some kind of frequency-dependent equilibrium. It'll go to this F star. And so here's what this kind of phase diagram is a function of alpha and beta that I've been talking about. And we're up here in the first quadrant where one has mutualism. And we'll talk about the third quadrant later. These quadrants here where alpha is positive, alpha is negative and beta is positive, or the reverse, those are Darwinian regimes. So this gray area is Darwinian. There's a simple selective advantage and so forth. And then similarly down here, I'll talk about mutualism for the next couple of slides. And then the last part of the lecture will be what happens when we're down here, where I have antagonism. Sometimes it's called competitive exclusion. I see that Shubha has raised a hand, please.
Alpha.
I'm sorry, I'm a little distorted. Could you repeat the question, please?
Am I audible?
Yes,
I can hear you now. If you speak slowly, I'll be able to get it. Go ahead.
What is alpha and beta in the equations here?
Thank you very much. So they're phenomenological parameters that tell you the boost in the reproduction rate of yellow, that's what beta does, when there are green organisms around. Similarly, alpha is a parameter that tells you the boost in the reproduction rate when there are blue organisms around. And so it's just a way to model the fact that yellow would like to have blue nearby because it can get tryptophan from it. And so if there's extra tryptophan it can get for free, doesn't have to make it itself, it will grow at a slightly greater rate in the presence of blue. And conversely, the blue will grow at a slightly greater rate than this background growth rate when there's yellow around. does that answer the question about the meaning of alpha and beta?
Yes, thank you.
Right. And while we're on the subject, thank you for the question.
Sir,
Yeah. Yep. Go ahead.
yeah, I just missed. Oh, G is a constant, right? Here, Wx equal to G.
G is assumed to be a constant, and it's the perhaps very tiny growth rate it would have in isolation. If I just put a yellow cell down on a Petri dish, it can't make tryptophan, but maybe it can get by without it, but it doesn't grow as well. So G is a background growth rate, and I just assumed they were the same for simplicity here.
Should not there be any dependence on only effects like the initial frequency of the the growth rate of the yellow cells should be dependent on the frequency of the yellow cells, right? Like in the normal
so the growth rate of the yellow will depend on the frequency of the blue, and the growth rate of the blue will depend on the frequency of the yellow. It's true that there could be other terms, and we could add them if we wanted to make the problem a little more richer and complex, where if the yellow, there's just too many yellows, then we run out of nutrients or something like that. So in a more sophisticated model, and I'll probably get to that on tomorrow, would have that effect, but I'm not including it now. The other thing I would say is that, you know, if they were poisoning each other, and we will get to this in about 10 minutes maybe, then alpha and beta could be small but negative. So in that case, the yellow would not want to have the blue nearby, and the blue would not want to have the yellow because alpha and beta could be negative. Then I'm down here, and that's sometimes called the competitive exclusion quadrant in this little phase diagram, or antagonism. So that's something that occurs in nature as well that we'll have to deal with. Any other questions?
Okay, sir. Actually, I just wanted to ask.
Go ahead,
like,
please.
Yeah, like in exponential growth, there the growth rate is growth rate is a function of x, right? Like dx is equal to kx.
I'm sorry. Can you? I'm not understanding. So can you please repeat the question?
Yeah, like when the in bacterial population, we see when the total number of bacteria is less and there is enough resource, then it grows exponentially, right? Where dx dt equal to kx, like that.
Yeah, that's true. Thank you. We are. That's a very good question. We're in a regime where it would be in a lab. It would be like a chemostat, where you have allowed the bacteria to have constantly replenishing nutrients, and they've all grown up to their carrying capacity. So, and so it's a powerful simplifying assumption, which is not always true. The initial exponential growth, we assume that's all saturated for both populations. And I'll write down equations that have that initial exponential growth in it. But here I'm assuming that, like as if it were in a test tube, that I have a fixed overall population, that the density of red or the density of yellow plus the density of blue is a constant. And so if I lose blue, I get yellow back again. And so we're not in an exponential growth regime here. We've grown out of that, and both populations, when they are present at their carrying capacity. And all we're looking at here is the relative proportions. But it's an excellent question, and we'll talk more about that tomorrow when I talk about the effect of compressible turbulent flows on organisms in the ocean.
Okay, sir. Yeah, another question which is might not be relevant here. So in the 2 slides report, the yellow and the blue mutualism. Can you go back to the slide? 2 or 3 slides.
So what do you want me to do? Sorry.
Yeah, go back 2 or 3 slides. Yeah, this one. Yeah, so if we culture these bacteria in a tryptophan-rich medium, so the yellow cells are getting enough tryptophan, right? They need tryptophan. They're tryptophan deficient, the yellow cells.
That's right.
And in a tryptophan-rich medium, but deprived of leucine, then the yellow cells will be getting tryptophan, but they have to spend some energy to make leucine, right? So
Okay,
in that scenario too.
Yeah, so let me answer that question by going to the next slide, if I may. So let's do the experiment. So I think this is going to answer your question. If it doesn't, please let me know. So here we have these two strains, yellow and blue, of yeast, spotted onto a Petri dish. But the Petri dish is it's rich media, and it's so rich it has leucine and tryptophan. So they don't need each other, even though they can't produce the amino acid themselves. And this is what happens over 6 days or something like that, 144 hours. So this is the yeast analog of the bacterial range expansion. And you see in this case, they don't care to stay mixed. Locally, because of number fluctuations, you get blue or yellow, like the spokes of a wheel. They don't wiggle as much as the bacteria did, probably because the frontier is thicker. And so the number fluctuations are not quite as small. But here we do have abundant media, and the fact that one strain, the blue strain, can't produce tryptophan and the yellow can't produce leucine is irrelevant. But now let's repeat the experiment on a medium where we deliberately have everything else that they need, but no leucine and no tryptophan. And let's look at the difference. And I think you can see that they stay mixed. They're not, it's sort of like arrested phase separation. If I call this thing over here on the left phase separation, here the phase separation is arrested, and there's a length scale that emerges, which is roughly, I think, how far the tryptophan or leucine that these guys are producing can diffuse in a generation time. So this is obligate mutualism. They have to stay mixed. So there's a important science here, and it's illustrated on the next slide as well, where what Melanie Mueller did was to start off the initial colony with different ratios of yellow to blue. So here there's 100 blue yeast in the initial inoculant for every yellow, and here there's 10 for every yellow. Here it's a 1-to-1 mixture. That's the movie I just showed you, basically going on in time. These are like frames from that movie. And then here the ratio is reversed. There's 10 times more yellow than blue. But look what happens. I get virtually identical final state. So this seems to reflect this mutualistic tendency to seek out a particular value of F star. And that seems to show up rather convincingly in these experiments. So that's what happens with mutualism. What I haven't told you is what is the effect of noise. And so a better version of this model here of what happens to the frequency. You can think of this as an equation for the frequency at the frontier. Suppose I'm neglecting inflation here I'm just looking at the frequency locally, say for an approximately linear inoculant. Here's this cubic polynomial. And now I want to add some noise. And so this is the noise due to the discrete number fluctuations. So this is a nonlinear stochastic differential equation, which is a rather new type, at least for physicists. I think some biologists like Nick Barton know already about this, but it's a nonlinear thing with this noise, which itself is nonlinear. And I'll just say that if I look at this first quadrant where alpha and beta are positive, the effect of these number fluctuations is to reduce the effect of the range where the mutualism takes over. So in this red region, the colony will remain fixed. This is a numerical simulation, not an actual experiment, although so similar behavior. And because of the number fluctuations, even though you might think in a simple mean field theory approach that they would stay mixed, in fact, yellow dominates here, blue dominates here. And there's a line of phase transitions actually between this mutualistic phase and yellow dominating and blue dominating. Now, one thing you can ask is how is it that blue dominates or yellow dominates? What is the nature of the Fisher wave that goes with this cubic polynomial? And that's what I'm going to talk about in another couple slides. So the summary is genetic drift, number fluctuations reduces the domain of mutualism. In the first quadrant, it's not the entire first quadrant, it's actually this little thing here with these strange pinched phase boundaries, which is kind of interesting. So any more questions about mutualism before I go on?
Yeah, I just have one quick
Please.
question. So then I would again look at F going to zero limit near the nose. So then it's basically like, if I ignore the, the stochastic fluctuation, it's like just the different S being S not F star, right?
Yes, that's right. That's right.
That's all that's all will happen, right? The speed will just change.
The speed will, thank you, great question. The speed will change because if we go through that little nose calculation, the speed will change. But as I'll show you, the wave is fundamentally different when the nonlinear terms are included. It's true, there is a wave, there is a wave solution, and we can get interesting properties of it by going to the nose again, just as we did before. But this gives rise not to the conventional Fisher wave, which is sometimes called a pulled wave. It gives rise to a new kind, which is called pushed. And I will tell you about the pushed wave in the coming slides.
Thank you.
Right. Okay, what about antagonism? Suppose we go to the third quadrant and make alpha and beta negative. And this is very recent work. The experiments were just published by really excellent postdoc Andrea Giametto, who's now on the faculty at Cornell University. And this is a classic case where we get also cubic polynomials, Kavita, and we get pushed genetic waves. So I'll focus on the antagonism because it's a little more interesting. And we'll see that now, because of the antagonism, to get a favorable mutation to take over, even if it's favorable, it has to occupy a bigger space in 2 dimensions. There's a critical droplet size, just like the nucleation theory that is beloved by physicists and chemists. And in fact, these experimenters, Andrea and Andrew Murray, and then the theory was done by Max Lebentovich, they were able to see this in the lab. That's what I'd like to get to here. And these antagonistic microbial strains, you can make them by having bacteria with toxin, antitoxin cassettes. They make peptides. These are, I guess, alpha helices. They're poison. They're poison in some fashion. Not your relatives, not your immediate relatives, not your twins, your clones, but maybe your distant relatives. But you have an antidote for it. There can also be local antagonism where you literally attack your neighbor and drill a hole through the bacterial cell wall, thus allowing the osmotic pressure to release and maybe explode your opponent, like exploding a balloon. And there's actually lovely experiments of this so-called type 6 secretion system at Georgia Tech by McNally et al., where they actually look at the coarsening and reveal a kind of line tension in the coarsening of these bacterial populations that exhibit antagonism. And my colleagues have actually managed to make a yeast analog of this with killer yeast viruses, Saccharomyces cerevisiae. So like the mutualism, they now secrete poisons. And I won't go through the details, but I hear there might be some biologists in the audience. You can correct me if I get some of the details wrong, but there's beautiful genetic engineering that goes into these yeast strains. This is budding yeast. It's budding out a daughter cell here. And what's inserted is a toxin that is controlled by a promoter that is activated triggered by the amount of galactose sugar in the system. And here's another promoter that makes another toxin that is activated by copper. And so what these very clever experimentalists have been able to do is to make a toxin pair, the kind of the mirror image of the mutualists. One is magenta, one is cyan, and they can control alpha and beta, which are now negative because it's a antagonistic situation. So in contrast to being in this quadrant, we're now in this lower left quadrant where they're trying to kill each other. And you'll see in a minute that there's a kind of line of first order phase transitions coming down here in the third quadrant. And on this side, everything's antagonistic, but on this upper side, the cyan will win eventually. And on this side, the galactose will win. And there's a kind of first order phase transition. They think of these as being spin up and this is being spin down with a line tension. So that's sort of where I'm headed. But what I think is really magnificent is we have an experimental system where we can explore these ideas. So this is the same kind of math that I've already talked about. I've just recast it in terms of alpha and beta. Here's the noise. Here's this cubic polynomial that's going to give rise to a slightly different kind of Fisher wave. And one way of recasting this that's useful, at least for some people, typically physicists, is to take this cubic polynomial and write it as the derivative with respect to f of a potential energy. It's not really a potential energy, it's biology after all. These are living active materials. It has the noise as well. But then, of course, this potential energy U of f is a quartic. And if I go back to mutualism, this quartic polynomial, as you might have guessed, depending as long as alpha and beta are positive, has a minimum. That's this preferred f star that I was talking about before. However, if alpha and beta are negative, here's the actual quartic potential. I just integrated it up. Then I have antagonism, and now there's a double well. There's a well here. It's only half a well because I don't go to negative f. That's unbiological. I don't go above 1, so it's only half a well here. I don't because I can't have more than 100% of, say, the blue organisms. But there's 2 minima, And then there's a barrier. So there's now a barrier. And that barrier is what makes this these push genetic waves so dramatically different than what would normally be the case. So here's just a change of variable. It's convenient to define alpha minus beta when they're both negative. This plays the role of a selective advantage. When you have antagonists, they can have a selective advantage for sure, but they also have a line tension, and that turns out to be related to the sigma parameter, which is the average of alpha and beta. And since they're both negative, this is a positive parameter, and it turns out to play the role of a line tension related to the line tension, and it plays the role of antagonism. And this is just another replotting of this double well potential. It's only half of a well over here and half of a well over here because there's a hard wall absorbing boundary conditions over here, absorbing boundary conditions over here. The system can never get past F equals 0. It can't go negative, can't go greater than 1 because the noise goes away at these absorbing boundaries. So if you think of a ball rolling down a hill, it has to stop here and it has to stop here. Yes, Nikhil, please.
Yes, so does that mean that in this doubled well potential, the initial conditions matter as compared to the previous case?
Absolutely, absolutely, great question. So up here, regardless of the initial conditions, you still get the same fraction of mutualism. Here, you can sort of see it. If I'm a little bit to this side of this 45-degree line, green will always win. But if I change the initial condition out here, blue will always win if I wait long enough. So that's the phase diagram. But now let me answer your actual question, which is if I start with an F that's a little bit to the left of this peak, I will roll down here. And if I start to the F, which is a little bit to the right, I will roll down here. The interesting question is if I set up a wave in space so that one side of the spatial region wants to go to this minimum, and the other side wants to go to this minimum, what happens? Okay, and that's an excellent question, and this is a fancy way to think about it. I won't go into the details, but it turns out let's neglect noise to keep it simple, and but look at the double well and look at it in one dimension. Just want to talk about one dimension. So if I look at it in one dimension, I can recast this whole theory in terms of a composite potential that has not only U of F in it, but also has this Laplacian. And I'll just let you, if you're no functional derivatives or otherwise believe me, that in the absence of noise, this df dt can be rewritten as a functional derivative of g with respect to f. And there's a kind of energy, composite energy that involves a gradient term in f, in the diffusion constant, and also this double well potential. And so here's the double well potential shown. Here's another representation of it. And as I change alpha and beta, both negative, as I change the relative proportion, the relative depths of these wells changes. And what you can show is that this g of t function has no choice but to decrease. And from the dynamical equations, I'm not showing it, it turns out that the g dt is this manifestly negative integral of df dt. And that means that if you have a little bit of seed of the lower energy state in the terms of U of F, it will find a way to grow and grow and grow and take over the system, just like nucleation theory. And this is an example in one dimension. And in fact, it turns out there is a solution of this cubic polynomial, an exact solution that was found by Nick Barton and his collaborators, much like the solution that we talked about and Kavita asked about in the foot for a conventional Fisher wave, but this is a pushed wave, and this is the exact profile. It's a relative of the hyperbolic tangent. Notice it's a function of x minus vt, and the velocity is selected and known exactly here. The velocity selection problem has been solved. It's convenient to work in a limit where small selective advantage compared to this surface tension term. And what you find in the language of Nick Barton is a critical propagule is required for a genetic variant to take over. And I just illustrated it here. This is totally different than a pulled Fisher wave, the one we talked about yesterday. Pulled Fisher wave, once it nucleates and escapes genetic drift, you can't stop it. It just goes and goes and goes. But here we have a Gaussian initial condition solving this differential equation numerically. And if the Gaussian is narrow enough, it dies out, even though the strain that is whose fraction is indicated by F, it has a selective advantage. So selective advantage dies out anyway because of the antagonism at the interface. That's one way to think about it. On the other hand, if I take the same shape initial condition and make it a slightly broader Gaussian, so B is 2.0 instead of 1.0, so this is the initial condition, the wave takes off and goes off to the right. It has a foot, just like Kavita was asking about, has an exponential foot. You can read that off here from this shape. And it asymptotically approaches this beautiful shape. And it's pushed because, remember, it depends on what F star is, the actual growth rate. And if F is greater than F star, it grows. If F is less than F star, it shrinks. And so that changes the profile. It sculpts the profile. And in a sense, it's driven from behind because only behind is F bigger than F star. And that's when this polynomial tends to drive F upwards. When it's less than F star, it tends to drive it downwards. Of course, they work together with this Laplacian term, and we get a classic pushed Fisher wave. So I was going to go through all this math. I'll spare you the math and just tell you the final results, which I think is particularly interesting, and it has to do with how I take this droplet critical propagule idea and transpose it to 2 dimensions. So 2 dimensions is significantly different than 1. So forget all this math. And the crucial thing is this equation down here. So you take here's a 2-dimensional version of this Fisher-like equation that I was talking about earlier. So I'll come up here and highlight it. This is the radial version. So I assume a spherically symmetric droplet, just to make it simple, and put in a wave-like ansatz and play around and try to get a formula for this dr dt. That's the radius of an expanding droplet. So here's a picture, cartoon of a little droplet that's expanding outward. And you go through this math. If the interface is reasonably sharp, the math is pretty accurate. And the final equation, perhaps more interesting, the actual derivation, is shown here. No noise at the moment, but what is it showing us? It's showing us that the rate of radial expansion of this droplet, if the droplet size is gigantic, so let me go down here and get my laser pointer back. If the droplet size is gigantic, this term can be neglected, and I just get a straight radial expansion at what turns out to be the velocity that I have here. So that's when the droplet's big enough in 2 dimensions, there's a critical nucleus, a critical droplet size much like classical nucleation theory, and this just expands and takes over. If delta is positive, delta is a measure of the selective advantage of, say, the yellow strain over the blue strain. But in 2 dimensions, because of this, the extra radial term in the Laplacian, there's another piece, which is minus d over r of t. So that says if the droplet size is too small, this growth rate is negative, and the droplet will collapse. So despite the fact that there's a selective advantage, the system will selectively favor the Darwinian preferred genotype will die out. So there has to be a critical droplet size. And the last thing I'll tell you about today is an experiment. Let's leave this out. But let me just say that if you look at this from this energy perspective, there's a droplet. If you work out the energy of this droplet of radius r0, these are the fluctuations, but let's leave those out. There's a term which is linear with a line tension. This line tension scales with the square root of sigma. And there's another term which looks like a condensation energy, just like the classic droplet theory of nucleation in the 1930s in physical chemistry and physics. The noise is different, so it's a new problem in that sense, but it's a kind of droplet picture. And this is a simulation that Max Leventovich did, where you simulate lots of selectively favored yellow droplets. If I blow this up, I get all these little yellow dots. But if I look at time 100 in the simulation, only a few of them have survived. The rest are too small. And they're less than the critical droplet size, and number of fluctuations maybe help them. And then later on, there are now well-established nuclei. They're all bigger than the critical size. And you can see this pattern of nuclei mimics the bigger growing islands over here. And so these have escaped the consequences of antagonism. And this is in physics, this is sometimes called the fate of the false vacuum. And in quantum field theory, it was popularized by Sidney Coleman, but Nick Barton read Sidney Coleman's summer school notes when he was a graduate student and a field biologist, and realized you could use the same mathematics for antagonistic organisms, which I think is a remarkable achievement. Anyway, experiment. This is where I will end. Experimental test. Is there a critical nucleation size? We could spot down selectively favored magenta killer yeast strains in a cyan background of different sizes, and look at what happens. And we're hoping that the big ones will grow and the small ones will shrink. And this is what was done. It was you spread these strains, and then you let them spread for a while, then you transfer them over to fresh media, fresh agar plate, and let them grow some more, and then each transfer is reflected by these pictures. And we want to see if something like this happens. Here is a compendium of many different experiments, and it depends on the droplet size. But if I start, if I look at what happens here as a function of droplet size, the droplet size is scanned from left to right. Time is going downwards in this picture. And you can see that here on the left, when the droplets are small, and I go through this procedure, in fact, the droplets die out. Here, they appear to grow. And it's biology, so the agreement isn't perfect. The droplets are not perfectly circular and so forth. But I think this nice experiment by Giametto and Murray, at least for me, convincingly establishes the existence of this critical droplet size. So to summarize, here we have this phase diagram, alpha and beta. Over here in these Darwinian regimes, we have poled conventional Fisher waves. they're virtually impossible to stop. Over here, we have one will take over the other if they're bigger than the critical droplet size. But once that happens, I have a pushed wave, which is a much more delicate thing and easier to stop, as we've already seen, and so forth. So with that, I think I'll stop. And thank you for your attention. This is a snowman in the Petri dish, which is appropriate in winter here in Boston, maybe. Any further questions, I'd be happy to answer them. So thank you very much.
Yeah, I just have one quick question. So if
Yes.
I include noise also in the pushed
Yep.
wave, in that case,
Yep.
I can ask for so the critical nucleus has to form. So one can ask for a sort of a establishment probability. Would it, does that?
Yeah, absolutely, absolutely. And there's a paper, a recent paper by Max Lerventovich and myself.
Okay.
There's more work that could be done, but we did the best we could to include the noise and include that establishment probability. But don't forget, Kavita, that the noise in this problem has this unusual scaling with the square root of f times 1 minus f, as well as 1 over n, 1 upon n. And so that means that where is f times 1 minus f largest? Precisely at an interface. So this is a very unusual kind of nucleation problem where there is an indeed establishment probability, but the noise, the temperature, so to speak, is hottest at the interface itself. These are systems which have hot interfaces. Well away from the interface, it's cold. And that has to be taken into account. And the best people have done so far, but undoubtedly could be improved, is to say, well, at the interface, I'll just take f to be a half. And so f times 1 minus f is a quarter. And then we have a constant noise problem. And then this looks something like something called Model A of Halpern and Hohenberg, Model A dynamics below TC. But that's just an approximation. And but yes, you certainly can talk about the establishment probability for a critical nucleus. And if you take a look at this paper with Lerventovich in, I think it's Physical Review E, in 2 years ago, we tried to answer your question, but there's certainly more work that needs to be done.
Okay, thank you. Are there any more questions? Okay, so I guess we can stop for today. And
All right.
see you
Thank
tomorrow
you.
again at 6:30.
Excellent. Thank you all. Thank you for the questions.
Bye-bye.
Bye-bye.